Recording in progress.
Why did it say that?
Can you tell us a little bit about the club and what the majors of the students that are in the club and how many are in the room with you?
Yeah, sure. So myself and Shalu are the co-presidents and the founders of the club. And Shalu is a public health major and I'm an animal sciences major. So we wanted to kind of combine both of our interests and start a club that really kind of advocates for the One Health approach and the intersection of animal science and human health. What else? Do you have anything to add?
That's the gist of it mostly, but like most of the majors in this club are like animal science and public health. So it's like a good like bridge between those two fields. So that's why I wanted to have you guys to talk about like the intersection between those two fields.
And do you have students interested in going to veterinary school or schools of public health, both?
Both. A lot of us are interested in grad programs. There's at least 2 of us that are pre-vet. And I'm hoping to also do a master's of public health in veterinary public health or veterinary epidemiology somewhere else if I don't go here. And then I know a lot of other people are looking to pursue like an MPH in different areas as well. Do you want to ask the next one?
Yeah. So the next question we had is, what is the most rewarding part or favorite part of the job for you guys?
I'll go first, I guess, again. One of the, I guess I'd have to say the most rewarding part of the job is, well, let me back up a little bit. We do both basic research and applied research at the lab. And the applied research may involve vaccine development or vaccine testing and evaluation or diagnostic test development and evaluation. And so I think for me personally, the most rewarding part of the job has been when you actually see your research being used somewhere or being used by someone, whether that's a vaccine that you worked on, whether that's a diagnostic test you worked on, or whether your research resulted in some kind of policy change by USDA or other animal health officials. I think that's very rewarding to see what you've done be used by somebody and hopefully advance animal agriculture, even if it's just in incremental steps.
I will agree with Mitch and try to come up with a different answer, but I like that there's this intersection of veterinary medicine and animal health and working with the public health sector. It always keeps it very interesting, and there's always a new problem or an incremental step to the next question that you might have. So it's always, well, not every single day, but for me, it's really interesting and exciting to be able to think about all these bigger problems or steps to solving a big problem.
Yeah, it's tough to follow up. Being an earlier career scientist, it's I haven't had necessarily the impact that Amy and Mitch have had over their careers. But I have seen, a couple incidences, and Mitch pointed out, if on the regulatory side, if they've taken into considerations, changes to regulations based on some of the research you've done, that can be rewarding because it's a way to see that, your research is having some sort of an impact as what, not just that you're doing it and it's going out there and it's not being used. And hopefully I get to, experience that a lot more. I so far think that's been a good rewarding aspect to it. But I also, I really enjoy the challenge of working with especially sort of an unconventional pathogen like prions, it's a protein. It's an approach that you have to take it's a lot different than maybe the approach that you would take with more of a conventional pathogen. And it's got challenges to it, and it's good to keep the mind working in that way, I guess.
Yeah, thank you all for that. It definitely sounds like a really rewarding field to work in. Our next question is, what is one thing you did as an undergraduate student that best helped you prepare for your current career?
I can go first this time if you want, because you guys steal all the good answers and everything. So,
And you are probably closer to that than us. We're having
Yeah.
to think further back.
Well, in undergrad, I guess one of the things that I started out by doing was, communicating and talking to different undergrad advisors I had or instructors that I knew, if they knew of any opportunities that I could maybe get involved with research. I didn't necessarily know that I wanted to go in research, but the science intrigued me to some extent. And so in one example, I basically ended up calling a scientist based on what my advisor said. And I ended up working in that lab for several years and that was actually during veterinary school. But once you're there, I think what really worked well for me is just to show that I'm open to learn as much as I can. I didn't know maybe a lot about what was going on and the different techniques they were using when I got there, but just trying to be coachable and open to different opportunities and just apply myself while I was there. I think that any scientist that's running a lab that's paying attention, runs their lab well, and has the heart of a teacher, will recognize good traits, and it should result in a good relationship that has a possibility to open up a lot of doors for you, whether it's continued work in their lab, getting into grad school in their lab, or even just letters of recommendation. And of course, learning different techniques that might be applicable to you, along the way. At the USDA, we have a program now through an interagency agreement with another department, and the program's called ORISE. And we have a lot of people that are in the post-baccalaureate, so they've gotten their bachelor's degrees. Sometimes they're between undergrad and graduate school, and they can kind of use it as an internship opportunity. And we've seen a lot of incidences where we've had people like this come in, and they end up being very successful applicants to grad school. So I guess, does that kind of hopefully gets to kind of the heart of the question of what you're asking about ways to get involved and do different things.
Why don't you go ahead, Amy? I'm still thinking, I have to go farther and farther back.
Well, I was just going to be general. I would just say try new things and try your best at everything, because you just never know what you might like or who you might need a recommendation from or cross paths with again in the future. And make sure you're well-rounded, because it's not just all about your coursework and good grades when we're looking for people to come work in our facility and labs. We like to see that you've been out there in the real world, tried things out, either had a job or been involved with activities and things like that.
In terms of things I did as an undergrad, I didn't know that this is where I would end up. And I actually had always wanted to go into practice, which is what I did after veterinary school. I went to Wisconsin and practiced for several years, mostly large animal. And that was really what my goal was in undergraduate and through vet school. But I did have the opportunity while I was in vet school to work in a couple of different labs, just as side jobs, and got exposed a little bit to research. So when a time came, when in practice, when I felt like I might want to explore other things, I did have a little bit of exposure to that, kind of like Amy is saying, to be open to a lot of things, to participate in diverse opportunities. Because as Amy said, you never know where this is going to take you. And it's actually quite a small world. So letters of recommendation and that kind of stuff, don't burn any bridges. It's a very small world. You never know when you'll run into people again.
The next question we had is, how did the pandemic affect your research and what precautions do you now have to take because of COVID?
I can go first since we'll take turns. We spent many months working from home because USDA had a maximum telework is what we call working from home in the federal government. And there were obviously things that we needed to go in to take care of in the lab and animal facilities, but everyone was told to stay home as much as we could. And as we went along, we started coming in kind of in shifts so that people could get into the lab and try to keep some research projects going. And my group of influenza researchers have been mostly in person, I don't for the last 6 months or so, but we wear masks. We had a brief period where we were able to come in without a mask on if we were vaccinated, but now we're back to wearing masks. And of course, we will be under the vaccine mandate for the federal workforce that the president has issued. So it definitely has been a very strange time, and we did a number of experiments. Mitch can and Eric can also talk about what they've experienced, but we just had to get creative with how to wear our PPE and respiratory protection and get in and out of the lab and animal facilities safely. So it's been a slower year than most, but we have been able to keep things going.
Yes, I'll go next. Yeah, Amy kind of described the policies and procedures at the lab. I in our group, I think we've pretty much all been there pretty much every day, all day in person for a year, maybe a little bit over a year. For me personally, the way how the pandemic affected me, I was actually 2 weeks away from retiring when the shutdown happened, and I got the opportunity to stick around and continue with my own research, but also get involved in some research with the COVID virus. And if you're in infectious disease research, that's kind of pandemic viruses don't come along, or pandemic diseases don't come along very often. And so if you're in that infectious disease research world, that's kind of a chance of a lifetime. So myself and Eric and some other scientists of the lab have been able to conduct some research, in different animal species, looking at their susceptibility to this virus. And so, it actually had a very positive effect on me and in my research.
Since we all work at the same place, there's not a lot different that I've really experienced as far as being here, the amount that we can be here and what goes on while we're here. But like Mitch talked about, there definitely were, I guess you could say, opportunities that popped up, and investigations that needed to be done about susceptibility of different agricultural species and livestock on whether or not they could get SARS-CoV-2. So that's been something that has definitely changed sort of the day-to-day research that I participate in, at least. So.
All right, thank you. Our next question is, some of you have kind of talked about it a little bit, but did you always want to be where you are right now? and what are some obstacles you had to overcome to get to the position you're in now?
I guess if we're taking turns, it's back to me to go first. I didn't know that I always wanted to be doing what I'm doing. Like I said, I'd always wanted to go to vet school and go into large animal practice, which is what I did. And I think everybody's experience in practice is different. I had a very positive experience, but I wanted to do something more, and started thinking about research. One of the obstacles I had to overcome were two. I had to go to my wife and say, I need to go get a PhD, so I need to go back to school. What do you think? And thankfully she was very understanding. And so to go into research, even with a doctor of veterinary medicine, I really needed an advanced degree. I needed a PhD. And so that's no trivial obstacle in both time and effort. I guess that's probably the biggest one for me.
Well, I did not have any idea I would end up where I am. I started out as an undergrad being pre-med, thinking I was going to be a psychiatrist. And then got interested in animal research and genetics. And so came to Iowa State in the swine breeding and genetics and an interdepartmental genetics program. But my dad is a large animal veterinarian, and I guess I must have had some sort of contagion from him, because I kept thinking about vet school and ended up there after all.
Yeah, it was really similar for me. I went down many different paths. I was originally planning on being a physician when I was an undergrad. I changed my mind about that maybe in the fourth year of undergrad. And so I had a year where I worked in an animal veterinary clinic before vet school, and took the GRE over that year and applied. But it was, kind of like we talked about before, it was, I had some research experience when I was in vet school. And so when I was out in practice, and I found that, I was convinced I was going to be a small animal veterinarian, and that's what I wanted to do, and a practice owner. And not that I ever really got turned off to any of that stuff, but I just had more of an interest and a curiosity in the pathophysiology of in the disease, of what was going on in the patients, that it made me want to go back and learn about that a little bit more. And the research exposure that I'd had in veterinary school, I think is kind of what allowed that to happen, and the right doors opened, was able to have that opportunity.
Hi, thank you for that. Would you guys be able to tell us a little bit more about like the specific research you work in? Like what got you interested in it, how it plays a role in public health?
Is it my turn to go first? Is that? Yeah,
It is, Eric.
yeah. So there are definitely aspects. So my research is in prions, or transmissible spongiform encephalopathy. So it's a group of diseases where you get degeneration of the brain, and it's fatal. And it's caused by a protein called the prion protein in your brain that misfolds and accumulates, and it starts to interfere with the normal brain processes. The one that most people are familiar with or have heard of is probably mad cow disease. And that one has obvious implications on human health, because when the mad cow disease outbreak happened in the UK in the mid 80s, and started there and went into the 90s, it contaminated the meat. And so people that consumed that, some of the people that consumed that, ended up developing a human prion disease called CJD. And so there's a pretty obvious a pretty obvious One Health correlation there that you can see between an animal disease and a human disease. And so a lot of the work that I do, that we do now, we work with some of the other prion diseases as well as besides BSC, like chronic wasting disease in deer and elk. And a lot of what we do is to test whether or not maybe different strains of these prion diseases could cross the species barrier if there's a possibility that humans could become infected. So I think that's the easiest way to think about what I do in terms of the One Health implications that it can have.
I work my first 4 or 5 years at the lab, I worked on a disease called brucellosis, which is a zoonotic disease, bacterial disease transmitted from mostly cattle, sheep to humans. And I worked at that time, we were looking at a new vaccine. And so my research involved evaluating some of the aspects of that vaccine, which went on to become the new brucellosis vaccine that's used around the world. The rest of my career has been mostly focused on bovine tuberculosis in cattle and some wildlife species like deer. So humans can acquire tuberculosis from a couple of different sources. Certainly, the most common one is from other people, but there's also a form that they can contract from humans, from cattle. And prior to mandatory pasteurization of milk in the United States, almost a third of the cases of TB in the United States were actually from cattle, mostly drinking unpasteurized milk. And so in the early 1900s, the USDA initiated a program to try and eradicate bovine tuberculosis from the US. And now we're over 100 years now in that program, and we still haven't eradicated the disease from cattle. So I work closely with the regulatory branch of USDA that pretty much runs the eradication program. And so our research is focused on improved diagnostic tests to help accelerate the eradication, and also the potential use of vaccines for both cattle and deer to try and add additional tools in the toolbox to finally get to that point of eradication. I've also had the opportunity during my career here to work briefly with other zoonotic diseases like cryptosporidiosis, which can cause diarrhea in humans, can cause diarrhea in calves and other neonatal animals, West Nile virus, and so I've had the opportunity to work on several different zoonotic diseases. Now the COVID virus is the first time I've ever worked on a disease or where there was a high risk of transmission from me to them. So that was a new aspect of that kind of research, a reverse zoonotic type of aspect.
So on that note, I work on a virus exclusively that can be transmitted from people to animals. So influenza A virus is a virus that can infect many animal hosts. And there's a kind of a special niche between humans and pigs. So there are some viruses that can easily move from people to pigs, like the 2009 H1N1, and then sporadic instances where the seasonal human influenza viruses can move into pigs. And then on the reverse of that, there's swine influenza viruses that are endemic in swine populations that can infect people. And when that happens, it's a reportable event by public health. So CDC is very interested in those viruses, particularly after 2009. And those are called variants, if you are familiar with influenza at all. So having a robust surveillance system in swine for influenza A viruses is really important to public health. And before 2009, we didn't have that in the US. So we worked closely with between USDA and CDC to get that program up and running. And it's been a highly successful surveillance system and lots of data coming from the swine populations. And the US has the best surveillance in swine for influenza of any country. So we definitely are interconnected with public health on that and work with CDC and influenza researchers on the human health side, as well as generate data that the WHO vaccine strain selection even uses at their biannual meetings for influenza vaccine viruses.
Thank you all for sharing that. You have like really diverse fields of research, so that's really interesting to hear about viruses, prions, and the bacterial diseases as well. And I know in a little bit, we'll also have a question more related to the influenza variants, but I think we have one last prepared question for you, which is, how do you practice self-care and manage your mental health in a field where you experience a lot of failures and loss?
That my turn. So I looked at your questions ahead of time, and I have I think for me, it's surrounding myself with good people, whether that's at work or my personal life, and making sure that you are engaged in really positive relationships. I also need to exercise regularly. That helps me with my stress. And the other thing I would just suggest is just not to take life or yourself too seriously. And that helps you kind of roll with the punches sometimes when you have to do that. And yeah, I think just appreciating what you have and the people that contribute to that is really important for me.
Go ahead, Eric.
Yeah, for me, it's important to take time, when I'm not here at work working or teleworking, at home working. But it was a little bit more difficult, I suppose, when we were maximized teleworking all the time. The line's a little bit more blurred about when you're working and when you're home. But taking time for family and hobbies, things that I enjoy doing, just to kind of recharge my batteries. Often find that when I've had a period of time, especially I take a little time off and do a hobby for that period of time. When I come back, I'm a lot more productive than and good to go again. So that's always kind of been really important for how I approach it.
Yeah, I would have to agree wholeheartedly with what Eric and Amy have said. All those same things, taking time for yourself, for exercise, for hobbies. For me, time with family has been very important and very helpful. And to be a well-rounded person. I really liked Amy's comment about surrounding yourself with good people, positive relationships. So many of the things for self-care and your mental health are pretty much similar to what a person would do anywhere in any kind of field. But it can take over your life if you let it. And you might always be thinking about work, or I don't know, you have to kind of sometimes be careful not to let it take over.
When you were talking, Mitch, I just was gonna make a comment that one of the great things about where we work is it's research, and at times it might be intense, but it's not usually life or death emergencies like you might be if you're practicing human medicine or an emergency veterinarian, and or in a public health emergency. So I think it's choosing the career path, you're going to have lots of options, and you have to think about where you thrive and where you are passionate. But for us, I think we can go into work refreshed and go home at night and kind of shut down for the most part, and until we start up again the next day. So it's been great for me personally to raise a family in this job, that I can make sure I'm spending time with my family and not working too many hours and never being able to shut it off.
Yeah, thank you all for sharing that. Mental health is one of the big focuses of our club, obviously as a public health issue, and then also knowing how high the rates of depression are in the veterinary field as well. So we really appreciate your advice. And then
I
one of our
will add one
questions.
thing that Amy reminded me of. When I left practice and came here, I started in October, and the first weekend I started here was Columbus Day, and that's a federal holiday, so it was a 3-day weekend. And I realized that was the first day off I'd had in a year and a half. So in practice, it's probably even more difficult. As Amy said, where we are, it's a little bit easier to keep things under control like that, but in practice, it can be really difficult.
Yeah, thank you. And then one of our questions is more directly for Dr. Vincent, but you both can chime in as well. And that was just, so with there being so many different variants of influenza and how quickly it's changing every year, how are influenza vaccines developed and how effective can they really be without knowing what we might be dealing with?
So there's, I guess, 2 lanes I could answer that question. There is on the public health side, how are human vaccines selected? And, I think for whether you're talking about the animal population or the human population, surveillance is really important for either of those, because you need to know what's circulating and when there are trends changing between different phylogenetic clades of whatever subtype you're interested in or focused on. So one of the issues with the human vaccine selection for the past year is that there was so low circulation of human seasonal virus during COVID because of staying at home and social distancing and wearing masks, that it was really difficult for them to have enough data to make an informed strain selection at the last couple of vaccine composition meetings. So for whatever you're talking about, either an animal vaccine or a human vaccine, you need to base the hemagglutinin and neuraminidase on what's out there circulating and evolving. So surveillance is really important, and then doing the laboratory assays to evaluate whether those HAs and NAs have changed antigenically, and then get those updated strains in the vaccine. So on the human side, there's basically one recommended set of influenza A viruses and influenza B viruses that go into the human vaccine. For swine, it's a lot more complicated because we have a lot more genetic variation in both the H1s and H3s. So for the US swine population, we have either fully licensed commercial vaccines, which just have a standard set of strains in them. But what's more common in the US is what we call either autogenous or custom vaccines. And so with those, a herd or a production system can find out what strains they have in their herd and have a vaccine made just to target their particular strains. So, so it's still very challenging because by the time the producer and their veterinarian tracks down what's circulating in the herd, recognizes that it's different from what was in their previous vaccine, and then contacts a vaccine manufacturer to get a custom or an autogenous vaccine. Sometimes the virus has already evolved further away than what the vaccine can be in the time that the vaccine can be manufactured. So it's very challenging for swine producers and veterinarians to keep up with what's happening with influenza in the pig populations. But I think we've made a lot of advances with the data that we've gotten through the surveillance system, and companies are doing a better job at being faster at making these custom vaccines. So we haven't won over the virus yet, but we're, I think, better off than we were 10 years ago.
All right, thank you. I know that was a very loaded question. There's a lot that goes into that. So thank you for answering that. And that's the last of our prepared and submitted questions. So now we're just going to kind of open it up to whoever has questions. If anyone does, if you want to just come up here to ask it so they can hear you, if anyone has questions.
So there was one question which, after Eric answered a little bit of it, I thought of some others. So Eric mentioned some of the opportunities that we have at our facility through a program called ORISE. And if you are interested in those, you can find those at a website called Zintellect.org, I think, or just Google Zintellect. But we also have some other USDA summer programs that are at different facilities, including ours, and that it could either be through APHIS or ARS, the research agency that we work for. And then there's also quite a few things that the CDC may offer, or schools of public health that have other either summer programs, or if you get finished with your bachelor's and aren't quite sure what to do yet, you can kind of have a bridging year to work and figure some of those things out. I did a CDC rotation when I was in veterinary school, which is how I got interested in influenza. So I think that's just one of the programs that CDC has for trainees.
Did you have a question?
Yeah. So, Dr. Palmer, I was wondering, what are the most common bacterial diseases that you see in cattle?
Well, I think probably some of the most common ones are things that cause respiratory disease, in whether you're talking about dairy cattle or beef cattle. Pasteurella is a big one. In dairy, mastitis is a huge problem. And you start to think about things like E. coli or Staph aureus, some of the streptococci. I think when I was in practice, respiratory disease, of course, I was in Wisconsin, and so we had an awful lot of dairy cattle, but respiratory disease and mastitis were huge problems. Now, you mentioned bacterial diseases. Oftentimes, when we're talking about respiratory disease, there's the bovine respiratory disease complex, and so there you have a mixture of viral and bacterial pathogens that kind of work together to cause severe respiratory disease. So many times the mixed infections, especially respiratory ones, are the most serious.
All right, thank you. Did you have another one?
Yeah, I had one more for Dr. Cashman. In your opinion, which COVID variant are you most concerned about?
make sure I unmute myself here. Um, I don't know why I got picked for this question. So I think, everything's, at least initially, this disease sort of has, split itself out into, risk stratification pretty clearly by age and any sort of uh, um, comorbidity that a person might have, or any other type of sickness that they might have. And so I think that, especially as you start to see waning vaccine antibody levels in people, that any strain could potentially be somewhat concerning to someone in one of those risk categories. I have less concern for people in lower risk category groups with low um, case fatality rates and low hospitalization rates, depending on the age category that they fall into. I don't know if that specifically answers your question, if you're asking it from sort of a human, kind of medicine standpoint. But one of the things that Dr. Palmer talked about earlier, and we'd used, some of the studies we were able to do here at the USDA, we looked at SARS-CoV-2 in lots of different livestock species, but the one that we found that it was able to infect were white-tailed deer. And now there's been a few other studies that have come out that have showed that they've found antibodies to SARS-CoV-2 in wild white-tailed deer, so that they were presumably exposed at some point. And the concern that goes into that, at this point, we're dealing with a virus. It's endemic. It's not going away. But now, there's potential, wildlife species reservoirs, in addition to, the cyclical spread that you get with endemic viruses just among human populations as well. So it's not going away by any means.
I'll just add on top of that, if you aren't familiar, there's a group out in Washington that maintains something called Next Strain. And they, I knew of them because they were doing influenza monitoring, but they are doing this for many different viruses now, including SARS-CoV-2. And so if you want to keep an eye on new clades or variants of SARS-CoV-2 that might be emerging, they've got a real-time phylogenetic tree and spatial analysis that you can keep an eye on that. And also, to piggyback on Eric's comments, is that's kind of one of the sectors that we all work in, is looking at these animal, either domestic livestock or wildlife reservoirs for these diseases. And if it does become endemic in an animal host, it will likely evolve away from what's circulating in humans if it's maintained. And so that is a reason to look for these animals, susceptible animal hosts, so we can keep an eye on that as part of the USDA.
Yeah, thank you both for talking about that. Obviously, COVID has kind of affected all of you in terms of research and everything. I also had a question. So I raised meat goats at home. So one thing that we kind of learned about through FFA and with working with the goats is that we're seeing, with using dewormers as heavily as livestock producers sometimes do, we can actually increase their worm load or increase their risk of infection. So have you had any experience with that, whether it's with parasites like worms or other types of infectious diseases?
So here at the NADC, as part of the prion research, we study scraping. And so we have some goats, but mostly sheep is what we do a lot of our studies in here. There's another ARS location that is working more with goats. But we have a breeding herd, and we do struggle with parasite management as well. And And a lot of times, especially the Haemonchus, the abomasal worm, is one that we really struggle with. So during the lambing season, we'll see the egg shedding rise and peak, while they're shedding. And Haemonchus can be really nasty. They can have a really high worm burden, and the animals will become anemic, and it can kill a ewe pretty quickly. And so, and especially, even the younger ones that have never been, exposed before, because that worm burden is so high. There are different, management practices that you can employ to try to help manage, longer, grass on the pasture and rotating pastures, and trying to, keep maybe the ewes, when they're shedding large amounts of eggs, off those areas where the animals are going to be grazing later. But we are finding that there's some resistance to some of the dewormers. And so I think that there are different recommendations now, as far as what anthelminthics you should use and in what combination, and whether or not you should dry lot the sheep after you've given them, so that, but you have to kind of be careful too, because if it's not done correctly and you're giving a resistant, antiparasiticide, or an antiparasiticide that a subset of the parasites are resistant to, the only worms that you have left are ones that are resistant. So those are the only ones you're going to be shedding on the pasture. So it can be a bit tricky, and I know it's a challenge. And I think that people that struggle with, especially Haemonchus, have a lot of frustrations.
Thanks for answering that one. We only have about 8 minutes left, so if anyone else had questions, feel free to ask. Otherwise, all right, I think that's all the questions that we had, unless did you have any?
No.
All right, I think that's all of our questions. So thank you so much for spending this hour with us and answering all of our questions. And especially with being in a different time zone and everything, we really appreciate your flexibility and being here with us.
Yeah, thank you. And
Thank you. Good luck with
best of
your
luck.
organization, your
Thank
club.
you.
Yeah. And feel free to reach out if you're interested in summer opportunities, whether it's with us or elsewhere. We can definitely point you in the direction of some resources. So best of luck.
Great, thank you. Have a good night.
Bye, everyone.